The chromosomal basis of inheritance. This is genetics part 2. So when Mendel was doing his initial experiments and when Darwin was coming up with his hypotheses about evolution, nobody knew where in the cell exactly inheritance was. And of course, chromosomes were suspected because of how they move during cell division, and the fact that chromosome number did decrease by half during meiosis. This led to the idea that chromosomes might be where the hereditary material was, because that fit with Mendel's model that individuals have 2 copies of each gene and they separate during sexual reproduction. So chromosomes were suspected, but there wasn't any evidence that chromosomes were actually where inheritance was. Early genetic experiments with fruit flies and plants found linkage groups, genes that were, or traits that were not controlled by the same gene, but whose alleles tended to stick together during meiosis and reproduction. And these were called linkage groups. And it was found that generally the number of linkage groups would be close to the number of homologous pairs of chromosomes. So for example, in fruit flies, there are 4 pairs of homologous chromosomes and 4 linkage groups were found for the genes, as many new fly mutants were discovered. And for peas, the species that Mendel used, there were 7 linkage groups found, and peas have 7 pairs of homologous chromosomes. So this was another piece of evidence that, in fact, it's the chromosomes, but it's not direct evidence. So how are Mendel's laws related to the chromosomes? So we talked about the law of segregation. Remember, I'm going to say law with air quotes because it's not really a law in the same sense that laws of physics are laws. We should call it the general rule of segregation because in biology there's exceptions to everything. So the law of segregation says that for those 2 copies of any trait that are on the pairs of homologous chromosomes, they're going to separate during gamete formation, and the gametes are only going to get one of the parent's copies of that trait. So we can see that here, that when we finally get down to the end of meiosis down here, there's only a single copy of each of the 2 traits here. So in this dihybrid cross, so a dihybrid cross just means we're dealing with 2 traits instead of just 1 trait, which I showed you a Punnett square for that earlier. So we have 2 traits here, yellow versus green seeds. Yellow is dominant, green is recessive, and round versus wrinkled seeds. Round is dominant and wrinkled is recessive. These are 2 different genes that control these traits. So the capital Y is for yellow and the capital R is for round. And then the lowercase letters are for the recessive versions of that trait. So for Mendel's law of independent assortment, so remember again, this is a law in genetics, more like a general rule, that Mendel deliberately chose traits that in a dihybrid cross sorted independently. And that's because almost all of them were on different chromosomes. 2 of them are actually on the same chromosome, but really, really far apart. So if 2 traits are on different chromosomes, when gametes are formed, we're going to expect to get all the possible combinations in the gametes. So if we have our gamete formation down here, we have gametes that have 1 copy of each dominant trait, 1 allele for yellow, 1 allele for round. We are going to expect gametes that have both recessive traits, 1 recessive green and recessive wrinkled. We're going to expect gametes that have 1 dominant and 1 recessive of each, so yellow and wrinkled, and round and green. And we will expect these to be equal. They're all equally likely if these genes are on different chromosomes in a dihybrid cross. We expect them to all be equally likely, and if you cross them together, you will get that Punnett square that I showed you in the previous lecture with the 9 to 3 to 3 to 1 ratio of phenotypes. If these are on the same chromosome, if if yellow versus green and round versus wrinkled, if those two genes were in fact on the same chromosome, we would not get an equal distribution of all possibilities here. In fact, we would get more of the parental types, which these would be the Mendelian parental types here, the two dominants or the two recessives. The first real solid experimental evidence that genes are in fact on the chromosomes, and now remember at this time DNA still was not known. It wasn't known that DNA, we're not talking about DNA because you remember the chromosomes are made up of protein and DNA. The chromosomes are about 60% protein, and they didn't even know about the DNA component of the chromosomes when T.H. Morgan started doing his experiments. So he was an embryologist, and he pioneered using fruit flies as an experimental subject. Fruit flies are ideal for a lot of reasons. They have few chromosomes rather. They only have 8 chromosomes, 4 pairs of chromosomes in the diploid adult. They produce lots of offspring. They're easy to handle in the lab. You can grow them just on this solidified yeast nutrient mixture in little glass bottles. They reach sexual maturity in only 2 weeks, so you can get a new generation every 2 weeks, which is really fast. Before we had any kind of DNA analysis, the fact that they had what's called polytene chromosomes made their chromosomes easier to study. They have the polytene chromosomes are multiple strands, multiple chromatids that stick together into these long and very detailed banded type of chromosomes that are really easy to study. Now this is less important because now we can just sequence the genes and see what exactly the DNA has, but this was really important before we had any of that. So fruit flies are very tiny. Generally they have red eyes. If you are in the US, you will see a few species of fruit flies that are all very tiny. You will mostly see them on your fruit when you don't want them to be there. The human chromosomes again, so human chromosomes are a little bit more complicated to study than fruit fly chromosomes. We have, again, 22 pairs of autosomes. So the autosomal chromosomes are the ones that everybody has. Everybody has 22 pairs of those. And then we have 1 pair of sex chromosomes. Now, before DNA analysis was available again, human chromosomes were studied in a similar way to fruit flies, and that was by banding pattern. But even with just that very kind of primitive method of looking at chromosomes, large deletions could be identified and identified as the cause of particular genetic disorders. So we talked about sex chromosomes a little bit already. So almost all individuals who are XX are female, and almost all individuals who are XY are male, and that's due to the activity of a specific gene on the Y chromosome during embryonic development. The SRY gene is on the Y chromosome. Now, the Y chromosome is the smallest human chromosome. It has around 200 genes, depending on what you define as a gene, and it's repetitive, so it's really only about 100 unique genes. And one of those genes is the SRY gene, and it's the activity of this SRY gene that sets off the development of an embryo towards male instead of female. So if the SRY gene is mutated and doesn't function, either isn't transcribed or is deleted, an XY individual will develop as a phenotypic female, but will be infertile, and that person would be counted as an intersex individual, which we'll talk more about that. So normally for, if we do a Punnett square here of sex determination, we have females are going, one of the sex chromosomes is going to go into each gamete. So for females, that means all of the eggs are going to contain one X chromosome because females only have an X, mostly. And for males, that means one of the gametes is, or 50% of the gametes are going to contain an X chromosome, the male's only X chromosome, and 50% of the gametes are going to contain the Y chromosome. So if we do a Punnett square, it looks like we would expect 50-50 boys and girls. But in fact, we don't get exactly 50-50. At birth, there's slightly more boys than girls. I believe the latest numbers I saw was about 104 boys per 100 girls born, so slightly more boys. However, by age 1, there's more girls, slightly. So why would there be higher mortality of boys? Well, it's probably due to the single X chromosome, which we'll talk about that a little bit more. So that's mammalian sex determination, is all mammals, dogs, cats, seals, lions, all use this XY system for sex determination. It's not the only system though. So birds use ZW system. So instead of an X and a Y chromosome, they also have sex chromosomes. They evolved completely separately though, and so they have different genes on them. And in birds, it's the female who has 2 different chromosomes. So females have a Z and a W chromosome. Males have 2 Z chromosomes. For many insects, they have a single X chromosome will develop as a male, and any number greater than 1 X chromosome develops as a female. So XX, XXX, 4 X's, that's all going to be a female, and a single X is a male. And this is very similar to what we see as well in the fruit fly, which we'll talk about. So for fruit flies as well, 1X is male and multiple X's is female. Other insects have other weird things like the haplodiploid system, which you can learn about if you're going to take the second semester of this course. So for insects, so bees, insects that are bees, wasps, and that group, diploids develop as female and haploids develop as a male. So for male bees, which are a very small percentage of bees, you might know they're just the drones, all the workers and all the bees that you normally see are all females. Males are only made once a year, generally for mating purposes. They don't do any work. So that with the haploid diploid system, you have different consequences for genetics and relatedness of individuals. So that's a whole nother story about social insects. Very, very interesting. Okay, so around 1910, T.H. Morgan is working. So this was several decades after Mendel did his work. Mendel, of course, lived and died and published his work without knowing how important his work was going to be later to other geneticists. Morgan, doing his fruit fly experiments, and he discovered many mutants in fruit flies. Almost all of them recessive mutations, and this was one. So he found a white-eyed fly among the flies in his lab, and it was, he was cataloging his new mutations. And so the first thing you do when you have a new mutation of something is you want to find out if it's dominant or recessive. So to do that, you do a breeding experiment, and he crossed it with a normal red-eyed female. And just like he expected for a recessive trait, the F1 generation were all red eyes. That's just what he had gotten with many other experiments when he discovered a trait that turned out to be a recessive trait. So all the F1 generation was red eyes, the wild type, just like we would expect. And then he let those flies, the males and the females of the F1 generation, interbreed. And yes, they were all siblings. let them interbreed, and then looked at the F2 generation, and he got the ratio he expected. He got 25% of the offspring had white eyes, just like Mendel got with his purple and white flower experiments. However, all the white-eyed flies were male. And the only way he could get females with white eyes is if the father had white eyes and the mother was from his F1 generation, meaning she was heterozygous. So why was this important? So here's this experiment. So he took his red-eyed wild-type female, crossed to his white-eyed mutant that he found, F1 generation, everybody had red eyes, just like you would expect for a Mendelian recessive mutation. However, in the F2 generation, all of the white-eyed individuals were males. And this was important because they knew that male flies have one pair of chromosomes that has a single copy, their X chromosome, which is not exactly the same as our X. And then male flies, as I mentioned, for insects, having a single X makes you male, 2 X's or more is female, and they have this other chromosome that we call a Y chromosome. It's like a little scrap of a chromosome, and it doesn't really function like a human Y chromosome, triggering male development. So, because you can remove, if it's missing, it doesn't matter. So here we have our gene for white eye, and very often for genes, the name of the gene and the abbreviation, either single letter or group of letters that's used to represent that gene, is a description of or related to the mutation. So that's why even though red eyes is normal, the symbol for the white-eyed mutant allele is W. And to symbolize the normal one, we use this plus. And that's something that you see a lot in genetics. That plus means wild type. That's what the plus is for. So if you see an allele that has a plus by it like that, they're telling you that that's the normal wild version of that particular gene. And then here are the W by itself, that's our mutant, and W is for white. And that's a type of nomenclature that we see in genetics. So sometimes we use not a single letter because obviously there's thousands and thousands of mutant alleles known. So we use groups of letters. This one has a single letter because it's really old in terms of genetics. All right, so here's his first experiment. So here's his white-eyed male. And this male is going to have white eyes because it only has a single copy of that gene. So even though normally we say, for recessive alleles, they have to be homozygous, well, males can't be homozygous for anything on the X chromosome because they only have one. So here's our white eye allele. Here's our normal female. All the eggs are going to have a chromosome with a normal wild type allele on it. So here's our F1 generation. The females are going to be heterozygous. And the males are all going to get the red eye allele from their mother because males get one of their mom's X chromosomes. Males get their dad's Y. They don't get their dad's X, they get their dad's Y. All right, F2 generation, we take the males and the females here and cross them, and now we have the male only has one X chromosome, so all the daughters are going to get their dad's only X. And so they're all going to get a copy of that dominant wild type allele, so we have no white-eyed females. For the males, however, 50% of them are going to get their mom's white eye allele. So 50% of the males are going to have white eyes. So this is typical for what we see for an X-linked recessive trait. The mom's a carrier, dad is unaffected. We're going to see none of the daughters affected, but 50% of them will be carriers, and 50% of the males will be affected. T.H. Morgan's experiments were the first real evidence that, yes, in fact, genes are on the chromosome. So because male fruit flies are like human males, they only have one X chromosome, males can express that recessive phenotype with only a single copy of the gene because they don't have another copy. So the reason females have to be heterozygous is because they have two, or have to be homozygous is because they have two copies of everything. So for a female to express a recessive trait, they have to have two recessive copies, one on each chromosome. And that's true even for the X, because females have two X's. For males, however, they only have one X. The Y chromosome is missing almost all the genes that are found on the X chromosome. So for almost everything on the X chromosome, males have a single copy, and we call that being hemizygous. So there's that word right there, hemizygous. Males only have, hemi means half, like hemisphere. So hemizygous means there's only a single copy of that particular gene in the genome. So for almost everything on the X chromosome, there's only a single copy in the male genome. So for traits that or for all the genes that are on the X chromosome that males have only a single copy of, if there's a recessive allele there, we're going to see it way more often in males than in females, because males only need one copy, females have to have two copies in order to express that recessive trait. There are dominant X-linked alleles as well, and in that case, because remember, if it's dominant, we're going to see it in the heterozygous form. So for females, they have two copies. If they have one dominant deleterious allele, they still have another copy on the other chromosome, and that tends to make these dominant conditions a little less severe. Males have a single copy, so for the dominant X-linked genetic disorders, although they affect males and females equally, males tend to be much more severely affected because they don't have a second normal version of that particular gene. So here's our what we get for X-linked recessive traits. So on the left here, so affected individuals are colored red. So those are individuals who have the phenotype for the X-linked recessive trait, whatever it is. Pink indicates carriers. So remember, for a carrier for a recessive trait, that means they are phenotypically normal, but they are carrying that allele to the next generation, even though they are not affected by it. So if the father is affected and he has the trait, that means it's that allele is on his X chromosome. So you notice the lowercase n there representing the recessive allele. The female here has two dominant normal alleles for whatever the trait is. And in that case, if the father's affected, none of the sons are going to be affected because boys get their dad's Y, not his X. Girls, however, all get their dad's 1X. So all of the girls are going to be carriers and the boys are going to be unaffected. Now, if mom is a carrier, dad is not affected. Now, 50% of the boys are going to have it because all of the offspring of this female, 50% of them are going to get the X with the normal allele, 50% of them are going to get the X with the recessive affected allele. So 50% of the daughters are going to be carriers because dad's got a normal allele on his X. Girls all get their dad's 1X. So none of the girls are going to be affected, but half will be carriers. 50% of the boys, however, are going to be affected. Boys get their dad's Y, not his X. So 50% of the boys are going to get mom's X chromosome with the disease-causing allele on it. Now, the only way we can get girls who have one of these X-linked recessive traits is if father is affected, because girls get their dad's only X, so it has to have the recessive allele on it. And mom also has to be a carrier because girls have to have two copies. So in that case, dad's affected, mom's a carrier. You will have daughters who now 50% of the daughters will be affected. And also 50% of the boys will be affected because mom's a carrier. So it's exactly the same as this situation over here for the boys. So that's why for these sex-linked recessive disorders, they are very rare in girls. Even though the allele prevalence in the population is the same, because most of the time when girls get the allele, it's in a heterozygous condition, so they're not affected. They're just going to be carriers. We're going to talk about 3 X-linked recessive disorders as examples. One is colorblindness, and that's red-green colorblindness. There are quite a few genes that contribute to our color vision. One of them's on the X chromosome. So that's why red-green colorblindness, that means that an individual who has that cannot distinguish between the colors red and green. Red and green look the same. And that's on the X chromosome, and so that's the most common type of colorblindness. Duchenne muscular dystrophy, there is one gene that is needed by muscle stem cells to create new muscle cells that is found on the X chromosome. So, and that is, there are many kinds of muscular dystrophy. They're all genes on the autosomes though, so they're very rare and they affect boys and girls equally. Duchenne muscular dystrophy is the most common kind of muscular dystrophy simply because this gene is on the X. It's recessive. Now you might wonder, okay, for these recessive disorders, are there cases where females being heterozygous are affected? And the answer is yes, and we'll talk about that when we talk about X inactivation. Hemophilia type A, there are many different kinds of hemophilia. So a hemophilia is means blood clotting is not occurring normally. Hemophilia type A is the most common kind of hemophilia simply because the gene for that particular protein happens to be on the X chromosome. Of the 13 protein factors that contribute to human blood clotting, one of them, factor 8, is on the X chromosome. So that's the most common type of hemophilia, and it's almost exclusively in boys. The same with Duchenne muscular dystrophy, almost 100% in boys. So here's what we would get with a Punnett square for one of these. So for hemophilia A, so mom is a carrier, so the capital H here represents the normal clotting factor 8. The lowercase h represents the defective allele for factor 8. So if you have half the normal amount of factor 8, that's perfectly fine. Your blood will clot normally. Male here who is unaffected. So in this case, 50% of the boys are going to get mom's chromosome with the hemophilia allele, and they will have hemophilia. The daughters who get that allele will not have hemophilia because they got the good copy as well from their dad. So they will be carriers, not affected. So be careful of that on test questions, whether the question is referring to someone who has the allele versus someone who has the condition. Because for females, just having the allele, they are not going to be affected unless they're homozygous. For males, however, because they only have a single copy, if they have the allele, they're also going to be affected. Probably the most famous family with hemophilia is the British royal family. And in this photo, we have Queen Victoria down here, surrounded by her children and grandchildren. There is the crown prince there, who will be the next king after her. She was queen for around 60 years until 1901, I believe. And one of her sons, her youngest son, had hemophilia. And so here's their very large family tree. Here's Queen Victoria here, and she had 7 children, I believe. 1, 2, 3, 4, 5, 6, 7, 8, 9. I misremembered. 9 children. And in this large family tree here, colored in individuals have hemophilia, and the half circles here are the carriers. And you notice all the carriers are females because males can't be a carrier for an X-linked trait, because if they have the allele, they're going to have it. They can't be a carrier for it. And there aren't any affected females on this tree. So that tells us for sure that this is an X-linked trait, because all the affected individuals are males in this case. And it might be, this family tree here might look a little bit confusing. You notice this little curve in the line here? That's because these lines are not connected. So that's showing you that this goes over. So Alice's husband here was from this extended family over here. Another sibling in this family married another daughter in the family. And you'll also notice something else here, and that is that Queen Victoria and her husband Prince Albert were first cousins. Queen Victoria's mother and Prince Albert's father were full siblings. So initially, people thought when Leopold was born with this genetic disorder, people often speculated that, well, it's, cousins marrying. But actually, that is not the case here. And it's believed that Queen Victoria was a new mutation, that a new mutation occurred in her because the royal family has extensive genealogy and medical records going back many generations. And if there had been, even though, medical practice wasn't what it is now, notes from the royal physicians have survived for many hundreds of years. And people often go back and look at those medical notes and diagnose them with a modern eye. And in this case, there is no indication whatsoever that there's anybody in Queen Victoria's line who had hemophilia before her son Leopold. So that's an indication that this was a new mutation in the family line. So for Leopold actually married and had one child, and we would know, even if Princess Alice had had no children, we would know that she was a carrier because all the daughters of an affected male for one of these X-linked traits are going to be carriers. And we can see here for daughters, they have a 50-50 chance of being carriers. So we know Alice was a carrier because she later had a son who was affected. And we know Beatrice was a carrier because she also later had two sons who were affected. The current royal line down here, here's the current Queen Elizabeth and Philip. Oh, you'll also notice that they are cousins as well. Prince Philip and Queen Elizabeth can both trace their ancestry back to Queen Victoria and Prince Albert, but no hemophilia in the current British royal family. So X-linked or recessive or dominant, how can we tell that? Well, there are dominant X-linked traits. So how do we know this isn't dominant? Well, remember, if we're looking for something that's dominant, every affected child has an affected parent. And we don't see that at all here. So, in fact, going back to the very first one here, Leopold had unaffected parents, affected child. And here, unaffected parents, affected child. Unaffected parents, affected child. So when you see unaffected parents having an affected child, that's recessive. That's what we're looking for to tell if something is a recessive trait. Red-green color blindness is an interesting story. We see a similar pattern here. So we can tell again that it's recessive because we have unaffected parents, affected child, unaffected parents, affected child. And it's X-linked because in this little family pedigree here, all the affected individuals are males. Now you do occasionally get a female who has red-green color blindness. It's just way less common. So color blindness, though, is pretty common in humans. It's way more common than those other X-linked traits that I mentioned. Duchenne muscular dystrophy or hemophilia, those are both very rare. And one possible explanation for that is that color blindness isn't really deleterious, really, unless you're a hunter-gatherer, and you need to tell whether fruit is ripe. Being, red-green colorblind now in modern society, well, maybe your clothes don't match. But other than that, it's not really deleterious. So possibly that was a hypothesis why it's so common. In fact, it's not common in other primates. It's, in fact, undetectable in other primates. So the question was, is it just common because of our modern civilized society? It's use it or lose it. Mutations accumulate, and if they're not deleterious, they just get propagated. So somebody went and looked at hunter-gatherers and asked the question, are hunter-gatherers colorblind? For modern populations in the US and Canada, it's about 8% of males are colorblind, which is quite high for a recessive genetic condition. So for hunter-gatherers, the percentage of males who were colorblind was that level or even a little bit higher. One population of hunter-gatherers was found to have 10% red-green color blindness. So, okay, so this is not something that has been lost recently due to civilization. So people ask then the same thing that we asked about cystic fibrosis and sickle cell. Is there some positive selective advantage going on here? Is there some kind of natural selection that is leading to an increase in red-green color blindness? Something that is a disadvantage for other primates perhaps was an advantage for early humans, and so it was selected for. And there is, in fact, a vision advantage in individuals who are colorblind, and that is that they are not fooled by certain camouflage. And perhaps this was an advantage to early hunters, which our humans and our ancestors have been hunting for at least 3 million years, going back to the Australopithecines. That's probably when we started to switch from scavenging to hunting. And individuals who are colorblind, the military has actually taken advantage of this, and individuals who are red-green colorblind are actually sought out as snipers because of their ability to see through certain kinds of camouflage that you might imagine involve colors. And see through, see movement in vegetation and where others might not see it, who have color vision. So that's a possible reason why red-green color blindness is so common, because it was actually selected for by natural selection during our hunter-gatherer days. Okay, non-disjunction is another one of those old-fashioned words that's kind of gotten locked in. So they used to call the separation of the chromosomes or separation of the sister chromatids during mitosis disjunction. We don't really use that word anymore. But when the chromosomes don't separate or the sister chromatids don't separate, we call it non-disjunction. And non-disjunction leads to aneuploidy. Aneuploidy, remember, is an abnormal number of chromosomes for that species. So aneuploidy means that there are chromosomes missing or there are duplicates of chromosomes, more copies than there should be for a haploid or a diploid cell. So here we see a chromosome or a cell that's going to go through meiosis, and it has two pairs of chromosomes, the pairs of homologous chromosomes here lined up for the first division. The homologous pairs separate, but one pair remains stuck together, and so two chromosomes go to one daughter cell, and the other daughter cell now has no copies of that chromosome. The second division, we separate the sister chromatids, and so we end up with half of the gametes are going to have an extra chromosome, and half of the gametes are going to be missing a chromosome. These gametes should each have two chromosomes because we started out with two pairs here, but instead we have, they're all aneuploid, and some have extra and some have one. So aneuploid gametes can go through fertilization, and if they are missing one of the chromosomes, then we call that condition a monosomy, or if we put it in the adjective form, monosomic zygote. So a monosomy is when there's only a single copy of just one of the pairs of chromosomes. A trisomy, tri is 3, tri would mean that there are 3 copies of one of the homologous chromosomes, where there should be only 2 for a diploid cell. There's 3. And don't confuse these with triploid. So if an organism is triploid, that's there's 3 of all the chromosomes, not just one of them. When all of the chromosomes are duplicated, then that's polyploidy. So poly means many. Humans are diploid and most animals are diploid, but we can have more than that, which we talked about a little bit already. So triploid would be represented as 3N, and that indicates there's 3 full sets of chromosomes. So for each homologous, instead of a homologous pair, we have a homologous trio. And tetraploid would be 4 of each of the chromosomes. And there are normal organisms that are tetraploid or more, octoploid, dodecaploid. This particular species of frog, for example, is octoploid and is perfectly normal. Sometimes polyploidy is a speciation event. For this particular frog species, there's a closely related species that's only tetraploid, and there are other closely related species that are only diploid. So clearly, sometimes this occurs and it ends up being a speciation event, especially in plants. There are a lot of polyploid plants, and that seems to be a cause of speciation often, or speciation meaning a new species forms that has double the number of chromosomes as the parent species. Triploids are generally sterile, so our domestic bananas are triploid, and that's why there's no seeds, because the seeds in a plant are the sexually reproduced offspring. In order for there to be a seed, there has to be a fertilization event, there has to be a zygote, there has to be the new plant embryo developing in there. So if those things fail, if fertilization fails because there's no pollen, you will not get a seed and usually not a fruit, like in the case of apples, that's the case. But for bananas, we have the failure of meiosis to occur, because in a triploid cell that tries to go through meiosis, the 3 chromosomes will line up together, they will try to cross over, and all 3 will try to exchange parts of their DNA, and when separation occurs, they all are attached and they end up ripping the chromosomes to shreds. So we do not get a viable offspring from tetraploid individuals. Instead, they are all reproduced clonally, or for plants, we often call that vegetative reproduction. So bananas are all reproduced by cuttings because they cannot produce any viable seeds, or domestic bananas can't anyway. There are wild bananas, but the inside of the banana is packed with large seeds because they are fertile. And they can go through sexual reproduction. So triploids are sterile. In the case of plants, that's no problem though, as long as humans are reproducing them vegetatively. Chromosome breakage does occur. We talked already about mutations, and a mutation can be as small as a single nucleotide changed in the DNA. That's a mutation. But there's also large mutations and structural changes to the chromosomes, which can have significant consequences for an organism. So a deletion means a large chunk is missing that should be there. A duplication means there's 2 repeated segments in the DNA. An inversion means a piece is turned around 180 degrees from what it should be, and a translocation means that a piece has moved to another chromosome. And all of these things happen as a result of crossover errors. So during crossing over, 2 non-identical chromatids are going to exchange pieces of DNA. Well, sometimes a chunk ends up missing. Sometimes a piece gets flipped 180 degrees and put back in. Sometimes one chromosome gets 2 pieces and the other chromatid gets none. And sometimes a piece breaks off and ends up sticking to another chromosome and getting inserted somewhere else. And although these are usually bad when they happen, we can see that these have happened during evolution, because when you compare chromosomes from different species, like if we compare human chromosomes to mouse chromosomes, we can see that large chunks of the chromosomes are extremely similar, but in other cases we can see that there has been duplications, inversions, translocations especially. So a deletion is a piece that's missing. A duplication means one chromosome has, or one chromatid has repeated sequences. And this could occur from a crossover error as well, as can a deletion. Inversions as well. The normal gene order is reversed for some segment. Translocation, a piece that contains some number of genes is actually found on a different chromosome from where it normally would be found. And now these last three here, duplication, inversion, and translocation, often don't cause any problem for the organism in which these first find themselves in. The problem would come when that organism tries to reproduce, because when meiosis occurs and their chromosomes line up gene by gene for crossing over, the genes aren't going to line up correctly. And that can result in further errors later on, especially in the case of inversions or translocations. We can get crossover errors where now none of the offspring will be viable from that particular organism. The most well-known aneuploidy is Down syndrome, and that's because it's the least severe and the most survivable of the autosomal aneuploidies. So individuals with Down syndrome, it's considered a trisomy. Individuals with Down syndrome have an extra chromosome 21. They have 3 chromosome 21s. Chromosome 21 is one of the smallest chromosomes and has few genes on it. It's not the smallest, but it's one of the smallest, and it's probably why this condition is survivable. But even for Down syndrome, the range, the phenotypic range for Down syndrome is quite large. And this is not well understood why this is the case. There are some individuals with Down syndrome who are extremely severely affected, who have heart defects and esophageal defects and other significant defects at birth that require surgical repair. Other individuals with Down syndrome never speak, are never able to take care of themselves, and toilet themselves and fix themselves a sandwich. They can't do a job. That's the severe end of the spectrum. The other end of the spectrum are individuals with Down syndrome who are low IQ, job-holding individuals who can almost live completely independently, can live independently in their own apartments if they have a little bit of help from social workers or relatives. So it's a very broad spectrum of functionality for Down syndrome. Now, there are a few other trisomies that are known, but they're more severe than Down syndrome, of the autosomal chromosome. So as we'll talk about the sex chromosomes, trisomies of the sex chromosomes are completely different matter. We're just talking about the autosomes. So the 22 autosomal chromosomes, there's only a few trisomies that are known to even survive until birth, with Down syndrome being the most survivable. Monosomies, meaning only one copy of any of the 22 autosomal chromosomes, is not survivable to birth. We don't know of any individuals who have that. So those conditions are embryonic lethals or fetal lethals. So we do know that aneuploidy is more common the older the mother is. Not with the age of the father, but with the age of the mother. And a very few trisomies, the few trisomies that are known besides trisomy 21, which is Down syndrome, are trisomy 13 and trisomy 18. These are much more severe than Down syndrome. So where are the rest of them? There's nothing specific about these 3 chromosomes that makes them more likely to go through non-disjunction. We think all of the chromosomes are equally likely to have non-disjunction occur. So why do we not see the rest? Where are all the rest of the trisomies and all of the autosomal monosomies? Where are they? Why don't we ever see those individuals? It's because they are not survivable. Having an extra dose of all of those gene products on those larger chromosomes is not survivable and not compatible with life. And this could partially explain why so many early conceptions ended miscarriage. Now that we can detect pregnancy really early with detection of tiny, tiny amounts of HCG, which is the hormone that's initially produced by human embryos, we can detect that much earlier now in the last 20 years or so, and it has become surprisingly clear exactly how many miscarriages there actually are. It's far higher than anybody suspected before we could detect these fertilization events so early, and it's been estimated to be approximately 50% of zygotes do not result in a viable pregnancy. So why does the incidence of aneuploidy increase with the age of the mother? It has to do with what is happening in the oocytes. So oocytes are eggs, the immature eggs. So in mammals, in females, those oocytes form and start to go through meiosis during the female mammal's fetal stages. So in humans, that's about 7 months gestation. So for human females, all of their oocytes enter meiosis when they are a 7-month fetus. So that means that they sit there crossing over prophase 1 of meiosis, which that's when synapsis occurs. So we have the pairs of homologous chromosomes coming together. We have crossing over of 2 of the non-sister chromatids. And they sit there in synapsis in meiosis 1 for however many years until that egg is ovulated. So they don't complete the first meiotic division until that oocyte is starting to be matured and getting ready for ovulation. So the month it's going to be ovulated, that's when it completes the first meiotic division, and it doesn't complete the second meiotic division actually until after fertilization. So if we look at this graph here, so this is incidence of Down syndrome per 1,000 live births. So this isn't a percent. We have to move the decimal point 1 to the left to get the percent of birth. So in the age of the mother here on the x-axis. So when the mother is under 35, or even if we look at under 30 here, the chances that a live birth will be a baby with Down syndrome is 0.1%. So that's 1/10 of 1%, really, really low. Even at 30, it's barely moved. But when we get to 35, that's the age at which doctors start to become alarmed. We are up to between 0.2 and 0.3% of live births. So still pretty low, less than 1% of live births, but 3 times the rate of 20-year-olds. By the time the mother is 40, we are up to 10 per 1,000 births, so that's 1%. So 1% of live births are going to have Down syndrome if the mother is 40. If the mother is 50, we are up to close to 10% of live births. And also, when the mother is 40 or 50, the miscarriage rate is much higher. At age 40, if the mother is 40, the miscarriage rate goes from 50% to about 90%. And that could probably be explained by additional aneuploidies, all those additional aneuploidies that are not survivable. So, but the good news here is that, even if the mother is 40, that, and there's a 1% chance. That's still a 99% chance that the baby is not going to have Down syndrome. But that's why. And it doesn't increase, the chances of aneuploidy don't increase with the age of the father, because for males, the primary spermatocytes are constantly going through meiosis and being produced, and stem cells are dividing by mitosis. There's no pause in meiosis 1 for spermatogenesis.